Hi, hello, welcome to a Physionic detailed study analysis where we're going to be going over study 193, which is a really popular study that just released as the time of this recording last month on taurine. So the amino acid taurine and its supposed effects on longevity, health span, senescence, stem cells, all kinds of different health factors. So it's a really giant paper that was published in the extremely reputable journal Science. And we're going to go over it. We're going to go over every last piece of data, or 95% of the data that's included in this study. So if you're interested in finding out how taurine supplementation, or taurine in general, just has a relationship with many different health metrics, then you're in the right place. This is it. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, so this is my bread and butter. I read studies, break them down, make them more digestible and understandable for you so that you can understand the experiments that they're doing and essentially be able to understand how studies are constructed and the conclusions that we can get from particular studies. I do these open study analyses so that people can, one, see the data for themselves, and two, make their own conclusions. That said, I usually get comments from people asking me to shorten the videos. This is not the type of video to be shortened. The whole point of these kinds of videos is to be as expansive and to be as detailed as possible. That said, I realize that some people don't have the time, so therefore I have a conclusions and takeaways section at the end of the video. So if you'd like to skip to that section, please feel free. However, please be mindful of the fact that you probably will miss a considerable amount of context which will be applied throughout the entire video. So those that go through the entire video get all the information, and those that skip to the end just get the most pertinent information. Okay, with that introduction out of the way, let's go ahead and get into this video. All right, so here are the topics that we're going to be going over. We have taurine supplementation on lifespan, obviously. Then we'll go over taurine on health span, and that's really defined by essentially just the healthiest you can live over time. So looking at markers for muscle, bone, body fat, inflammation, and when I say more, I mean much more. So we're going to be going over a lot of that. Then we'll also be going over taurine and its relationship to our stem cells, and I'll explain why that's important later on, as well as cellular senescence, so kind of the opposite of stem cells. And then this is a lot of this data comes from animal studies, so the researchers wanted to find a relationship to humans, so they end up including some data in humans as well. So we're going to be going over some data on humans. Then for my insiders, that are part of the insiders community, insiders program, you also get access to some more data looking at humans, exercise, and taurine, as well as direct evidence of supplementation in humans, as well as the optimal dosing, which is actually going to become pretty important and pretty clear in just a little while as we get into the meat of the data. So the study, and if you're interested in getting to joining the insiders, then please feel free. You're always welcome to. It's linked for you, so you can just click on the link and then join there. Okay, so for this study, which is, like I said, it just released, it was I think completely public funding from a number of different grants by the NIH and different organizations that are associated to the NIH, so the National Institute of Health. And you can see that there's a lot of researchers that were involved on this study. And there's a good reason why it got accepted to Science, because they do so much research just for one paper. It's pretty incredible. Okay, so this is the paper. And you'll find notes and stuff like that linked as well for this paper. All right, so what are we talking about? We're talking about taurine, which I'm gonna show you taurine as this molecule just for simplicity's sake as we go through the presentation. But in its chemical structure, it looks like this. So it's got a nitrogen group, obviously, because it's an amino acid. Now, an amino acid is part of the protein family. So if you were to consume proteins, those proteins are made up of amino acids. And taurine is one of them. And it's kind of waffles between being an essential and non-essential amino acid. It really depends on your nutrition state as a whole. But usually these kinds of amino acids are considered what's called conditionally or semi-essential amino acids. So that's what we're going to be focused on. So let's get into the meat of this data. So it's not just that they look at animal studies, they do look at some human data as well, just right from the get-go. So in this experiment, all they're doing is just measuring blood levels of taurine, so the taurine molecule being found in the bloodstream, and relating that over age. So we've got taurine on the vertical axis, and we've got age on the horizontal axis. And obviously, as we move further to the right, the mice are getting older. In this situation, we've got mouse, we've got monkeys, and we've got Homo sapiens, whatever those are. That's a joke, I'm aware. And we see that taurine levels, regardless of which species that you look at, there's a decline in serum or blood taurine levels. So this creates this relationship of, okay, as we get older, we see reductions in taurine levels. So this is just an association, but you're still able to make that strong association of, okay, as age increases, taurine levels decrease. And it's not just like, it just happens in mice. It doesn't just happen in monkeys, which here they show it a little bit differently. They've got young monkeys of 5 years old, and then they measure their blood again 10 years later in 15-year-old monkeys, and they're able to tell, okay, there's this huge drop in the amount of taurine. And we're not even looking at percentages here. We're looking at absolute values. This is a huge drop in taurine. And then, of course, the most pertinent would be to look at us, looking at humans, unless an alien is watching this, in which case it might not apply. But for the most part, everybody else that's watching this, if you're a Homo sapien, which you are if you're a human, then you can see that at age, 5, 0 to 5, taurine is really high, and that's pretty understandable because taurine is heavily involved in our neurodevelopment and our general development as a whole. And it declines pretty rapidly. I mean, pretty steadily, but pretty rapidly regardless. So by age 20, you're looking at maybe 40% reduction in taurine. And as you keep going and going and going, eventually your taurine levels are incredibly low, like they would be with the monkeys. And the same is exactly true for the mice as well. So why does that matter? Well, it matters because here's where we can actually get into the data of what happens when we actually supplement with taurine. So if we supplement with taurine, and now we're going to get into animal models, and then we'll return back to human models, just to be clear. So most of the data, and I'm going to point out exceptions, but most of the data is going to be in animal models because we can actually manipulate animals. We can, not that it's the prettiest sight, but we can actually sacrifice animals to get the data that we need. We can't sacrifice humans to get the data that we need. So we're going to be using animal models. So for the animals, what they've done here is they've taken mice and they've either fed them the vehicle, which is a placebo, which is a control, which is essentially no taurine. So the blue is animals that were not fed taurine, and the red or orangish, let's just call it red, were supplemented with taurine. And then all they did was just measure how long it took them to die. So this is specifically looking at lifespan. And what they found is that, the way to read this is you've got 100% survival, meaning that all the mice are alive, right? And as you keep moving to the right on the horizontal axis, you have a longer and longer lifespan. So their age of the mice continues to grow. Now, what's really fascinating about this, and what's really exciting, frankly, about this, is that the animals are fed taurine not at the beginning of their lives, not at the 8-week mark, which is kind of the beginning of their adult life when you're talking about mice. or kind of, I suppose you could say like adolescence, they're being fed at the 14-month mark. So they are well into middle age and then fed taurine, and then they see the effects. So this is really remarkable. It's not one of those situations where you're like, oh, well, you should get ahead of the curve as early as possible because you might not see an effect later on. What they're showing here is that even if you take it much later in life, that you can see these effects, at least assuming that this translates to humans, which we'll get to that. And what we see is that with the animals that don't supplement with taurine, you start to see that the first one dies at, let's just call it like 21, 22 months. The first one dies, so you're down, or first few die, so you're down to like, let's say 90% survival rate. But the ones supplemented with taurine are still alive. So it's not until, at the point of like, let's say 15% of the animals that did not supplement with taurine had died before the first few have died with that supplemented with taurine. And overall, all the mice end up surviving longer with taurine compared to the non-taurine supplemented group. And they've separated it by sex. So they've got the female mice here, they've got the male mice here, and the results were consistent. And to quantify that, they measured it at about 10 to about 12% increase in lifespan, which is pretty substantial. Now, other models that they wanted to look at was C. elegans, which are worms, and yeast. What they found is that yeast had zero effect. So when they apply taurine to the yeast cells, there is zero effect. So it seems to be only specific to mice, and they did see an effect with the worms as well. So, but obviously the most important, or the greatest of the greatest interest to us, is probably going to be the mice as a mammalian model. All right, the next area of interest was to look at body fat, or body weight and body fat. And here they're separating it out across three groups. So again, they're feeding the mice at the 14-month mark, which for mice is about middle age, maybe a little bit older than middle age. And they've got the T0, which is these unsupplemented mice. These mice do not get fed taurine. And keep in mind, everything else about these mice is the exact same. So they get fed the exact same foods. They're in the exact same cages. Everything is the same. So the only difference between these mice is taurine supplementation. And then they've got a group that consumed 500 milligrams of taurine and another group that consumed 1,000 milligrams. So they've got a dosage, separation, and they've got two conditions with taurine. And what they found when looking at body weight, so this is the point of supplementation, and this is over time what happened to their body weight. And we can see that there's a non-significant difference between the unsupplemented and the 500 milligram condition. So this would imply that there might be a dosage effect, so that if you're consuming more taurine, that you're more guaranteed to have an effect. Now, I would argue that, and it's tough to tease out here, but I would argue that most likely if this had continued, that they probably start to see a difference. So I think the big takeaway here is that with lower taurine dosage, there's probably still an effect. It's just not as dramatic of an effect. But as it stands, we'll just go with what the statistics show, which is no effect between the low taurine and the no taurine, or control. Now, on the other hand, the comparison of the placebo, or the T0, versus 1,000 milligrams, we do see a statistical significance. So over time, essentially what taurine is doing is reducing body weight gain. That doesn't mean that it stops all body weight gain. It just it blunts the effect of body weight gain. Another measure to look at is to look at fat levels, which is probably more important for a lot of people than their actual body weight. And what they found is that there were reductions in body fat for both taurine conditions, which is really impressive. The next one is to look at some markers that most people usually don't really think of until they're probably in their 60s, 70s, and 80s, especially in their 80s, and that's bone mass. So, I'm not an expert looking at images of, or histology or anything of that nature of bones because I have zero training in that regard. So I'm just going to be showing you the images, and and I imagine unless you're let's say a pathologist or somebody that's in this field, in this specific area, you're going to be able to look at this, but otherwise, for the rest of us who don't have the qualifications, we're just going to have to base things off of the numbers that they end up reporting. So what we're looking at here is vertebrae. So the vertebrae that are taken from obviously the spine, and then here we've got the femur, which is this part of the leg right here. That's why I've got these arrows pointing to each one. So we've got the T0, again, these are the controls, so not given taurine. Then they've got the 500 and 1,000 milligram conditions of taurine consumption. Again, these images, it's difficult for me to actually be able to tell if there's any difference or not. So I'm just going to be basing things off of the numbers that they report. So they're looking at bone volume, and they're finding that there's a pretty substantial increase, especially if you look at these what are known as standard deviations or standard error of the mean, that there's a substantial, I should say, I suppose, maintenance, because I don't think that these animals are actually building more bone. It's just that they're able to maintain their bone mass much better. So, and you got to think the power of being able to maintain bone mass in your vertebrae. Think about that. As we age, we shrink. I mean, not by much, but over decades, it adds up. Imagine being able to maintain your bone mass, and obviously the cushioning between the bone mass is another issue, which they did not test. But still, this is pretty incredible to see such a huge increase. I mean, just think about the percentage increase of 3.26 to 5.17 or 5.49. That's over 50% increase. That's huge. That is huge. Okay, so over here, it's a little more obvious in terms of the actual bone mass or bone volume. We can compare this one versus especially this one right here. You see that these areas are much thicker. You see that there's some bone that's being interspersed between these areas. So, and you can see that the difference between 4.9 to 7.84 is huge. Again, well over 50%. We're talking like 70, 80% improvement in bone. So that's the actual bone volume. But another test that they want to do is to actually look at the skeleton strength. So they're looking at the maximum load that the femur can actually hold on to, and without incurring any sort of damage. And they find that it is improved. So the higher it is, obviously the better. And they find that it's a substantial improvement with taurine supplementation. The other one is to look at the fracture load. So I'm assuming this is the point where the actual femur actually fractures. And they find that, again, they can load the femur much more to actually before they actually start to see an impairment in the femur to the point where it's actually breaking. So this is incredible. The next one would be so they look at a number of different metrics. And again, for some of these, we kind of have to trust that these have been established methods. So I've never tested anxiety in mice before, but there are a whole slew of different psychological tests that have been assessed and reassessed for reliability and accuracy. So I'm just trusting the researchers that they know what they're doing. That said, neuromuscular coordination is essentially just your ability to maintain balance, be able to move effectively, being able to grab onto things, being able to move with dexterity, essentially. And what they find is that according to these different measures, so latency to fall, distance, so distance traveled, and total hanging time. So usually they do a bunch of tests where the animals are hanging off different objects. Sometimes it's a cage where they're literally hanging off something and then they have like a pillow below them. But obviously the animals don't know that. So they're just holding on for dear life. Well, all of them were improved except for the latency to fall with the lower taurine supplementation. So again, the dark blue is the high taurine supplementation. Light blue is the lower taurine supplementation. Grip strength improved with both conditions. Anxiety decreased quite substantially. And then looking at memory they're also seeing that there's an improvement. Usually you do some sort of maze test or some sort of recognition test for this. I've seen a few experiments on this before. So they see that memory was improved. Now, if you're reading this, you might say, oh, wait, the dark blue is lower than the light blue. That's a non-statistical significant difference between the two. So you would actually read that as they are both the same. So there's no difference between those two, but there is a difference between the two of them and the control condition. The next one would be, obviously, this is super important for a lot of people, is to look at glucose tolerance. So we're looking at, it's a proxy for like insulin resistance, insulin sensitivity. And here they're looking at blood glucose levels as they fed these animals glucose, a set amount of glucose, just like you would get for an oral glucose tolerance test. And that's what they're doing here. So you see, obviously, if you feed animals, if you feed yourself glucose, sugar of some sort, then you're going to see a huge spike in blood glucose levels. And that's what we normally see. Then the next goal here is to figure out, well, how quickly is it cleared out of your system? And after supplementation, they found that with both of the taurine conditions, that there's a faster decline in blood glucose levels. I don't think it's that substantial. It's not a huge decrease, but it's definitely present, especially at the 30-minute mark, and potentially getting almost to the 60-minute mark. And then they're looking at the area under the curve. So the area under the curve is the whole area under this curve right here. And they find that there is a substantial reduction in total amount of glucose exposure. So the glucose exposure in the bloodstream here. So the faster you're able to remove it, then the less time that blood glucose is in your bloodstream. And that also implies that you're more insulin sensitive. And then looking at fasting glucose levels, they see that there's a reduction in fasting glucose levels. And then they've got another measure here. So from percent of initial. And here it's really difficult to tease out, but there's a slight decline, a slight decrease in blood glucose levels in the two taurine conditions at the 15 and 30-minute mark. And then ITT is another measure of glucose. Essentially, we just see a decrease again in this condition. So across all the metrics of blood glucose, fasting, oral glucose tolerance test, etc., we see that there's a decline in blood sugar levels. Then the next one is an interesting one. It's not something that I'm very familiar with. It's not something that I've seen on a regular basis, but the GI transit, so gastrointestinal transit. So we're assuming here that the faster that food is able to get through, it's a better outcome, which is an assumption on my end. That may not be the case, but considering everything else has been positive so far, I'm just assuming that, and that may be a bias on my perspective. So I'll add an amendment to this video if that's not the case. So we see that there's a decline in transit time. So the food is readily able to be absorbed and then removed or put out of the system. So you poop it out. Now, the main one that I was interested in, and I think a lot of people are interested in, is looking at immunophenotype. So they're looking at a variety of different immune cells. So they've got white blood cells here, they've got monocytes here, and they've got granulocytes. So monocytes and granulocytes are a specific type of immune cell, and white blood cells are more of a class of immune cells. And to be frank with you, usually when we look at this, it's a little difficult to then assume that the differences here are actually meaningful, that just because we see differences doesn't actually mean anything. We'd actually have to relate that to something. And usually you have to look at other studies to indicate, oh, well, if you see a reduction in monocytes, then that's a good sign because you maybe have reduced inflammation, something along those lines. And that's usually the assumption. So what we find is when we look at white blood cells, that with taurine supplementation, either one, we see a decline in white blood cells. So obviously you don't want to eliminate all your white blood cells. You just want to reduce them to the point where you're not overactive on your white blood cells. Another one is monocytes. And what's really intriguing here is that specifically looking at monocytes, which are these circulating innate white blood cells that are found throughout your system. And when they're activated, they'll invade into your tissues. Let's say you get sick or something like that. They'll invade into your tissues and differentiate or turn into what's called macrophages, which you may be familiar with. These macrophages will then have different gradations of activation, so that you have quote unquote inactive macrophages. Not exactly right, but we'll go for it for simplicity's sake. Inactive macrophages going to then activated macrophages. And there's really a gradation between the two. So monocytes, here we see that there's no effect with the smaller taurine dosage, but there is an effect with the higher taurine dosage. And the exact same is true for granulocytes. Granulocytes are actually these right here, where you have, just as a side note, these immune cells have these granules inside of them that are damaging granules, and they're sequestered within the cell. And if there's a stimulatory event like a pathogen or something along those, like a microbe or some sort of bacteria or whatever, then these granulocytes will undergo a process called degranulation. If you've never seen a video of it, it's really cool. You see the granules inside, and then they just release all the granules to essentially, it's like a nuclear explosion that releases into the surrounding environment to essentially attack the microbe. It's such a wickedly cool phenomenon. So I certainly encourage you to check it out sometime, but maybe after this video. Okay, so what we see is that for the most part, having larger taurine doses seems to see a shift in this immunophenotype. Okay, we're going to get into the senescence. So senescent cells are essentially cells that have stopped replicating. They've stopped dividing because the cells are essentially trying to protect from becoming cancerous. So our cells have many, many different mechanisms to essentially stop them from becoming cancerous. One of the most common mechanisms is called P53. P53 is an anti-oncogenic gene or protein that essentially stops other proteins from driving cancer. And senescent cells will change their expression levels of particular genes, of particular proteins, to stop the cell. To essentially, they express more of the non-dividing proteins or enzymes that stop division. So they go from this dividing cell phenotype to this isolated cell phenotype where they just essentially shut down. But they're not dead. They're just shut down from dividing to essentially stop cancer progression. Then on top of that, the problem is that they can also release immune factors that recruit immune cells to then get rid of those senescent cells. Now, that's not a problem when you have a few senescent cells, because the immune system comes in, deals with it in the colloquial, the correct canonical pathway. That's fine. However, when you start to build up many, many senescent cells, then you have all these senescent cells that are screaming for attention by the immune system, and the immune system simply can't handle it. So you have this overactive immune system, more and more senescent cells, which actually convince other cells in their surrounding environment to also become senescent. And therefore you have substantial problems, because then the entire tissues are starting to become less and less functional as more and more cells are becoming senescent. That's why they're also called zombie cells, because they will, quote unquote, infect other cells. They don't actually infect them, but they can turn a perfectly healthy cell and essentially, quote unquote, molecularly convince it to become senescent. And that's done by these proteins called SASPs, or senescent associated proteins. Senescent associated secretory proteins. I knew I forgot a word there. Okay, so that's what we're looking at here. So one of the proteins that is highly upregulated in senescent cells specifically is this SA, senescent associated beta-galactosidase. So SA beta-galactosidase, if you see high levels of this particular protein being expressed, then the cells are most likely becoming senescent. So what are we looking at here? I'm adding these images here because we have, on the left side, we have the non-taurine supplemented, and on the right side, we have the taurine supplemented. And the way the beta-galactosidase occurs or shows itself is by blue. So if you see more blue, then you have more senescent cells or more senescent pockets. So you're comparing the brain here versus the brain here, the liver here versus the liver here, the fat here versus the fat here. And we can see, at least by my eye, and you would have to determine this for yourself, I can say at least with pretty good confidence, the liver and the fat, that there's this huge distinction in the amount of blue that's found in the liver versus the amount of blue that's found in the fat. The brain, not quite as much. I think that the effect isn't quite as potent, but it seems like there is a small effect at least. Now, to quantify this, so this is just a single image. So usually researchers don't just show a single image and they're like, oh, see, proof. Usually they're quantified across multiple measures and across multiple animals. So in that situation, they've quantified it here: brain, liver, gut, muscle, and fat. And they found that across the board that there's a drop in this senescent signal of beta-galactosidase. So this is really, really cool data. Okay, now we're shifting gears a little bit, and I'm going to explain this. You might be wondering, why are we looking at fish, Nick? So we're looking at fish because fish are another model organism. So we've looked at mice, we've looked at monkeys, we've looked at Homo sapiens, us, and now we're looking at fish. So we're looking at fish, specifically zebrafish, because they are a common model organism that's used in research because they're easy to manipulate. You can manipulate their genes quite easily. Oh, we also looked at worms, which is another one that we can manipulate their genes really, really easily. And they're faster to use for different genetic manipulations. So what am I talking in this situation? So yes, of course, you might be thinking, well, how does this apply to humans? Well, there's probably not a direct relation. It's not like we can say, oh, this affected zebrafish by 13%, therefore it will affect humans by 13%. The point is, can we see an effect across multiple model organisms? And if you see it in mice, if you see it in worms, if you see it in monkeys, if you see it in all these different model organisms, the odds are pretty good that something is going to happen in humans as well, even if the effect size might be different, et cetera, et cetera. So just to explain why they're using fish, it's just an easy model organism to use for this genetic manipulation that they ended up doing. So the genetic manipulation is SLC6A6. So they create a knockout. So they're essentially taking the gene that encodes for the protein, the transporter of the taurine transporter. So for your cells to take up taurine and actually be able to use the taurine that comes from the bloodstream into your cells, they have to actually enter your cells, right? Well, the only way they can do that is through this taurine transporter. And that's true for other molecules as well. For example, glucose. If you were to knock out the GLUT transporters, you would have extreme difficulty getting glucose from the bloodstream into your cells, and you'd probably die pretty soon afterwards because you'd be hyperglycemia through the wazoo. Now, with taurine here, they're knocking out the taurine transporter, and that's what we see here. So the SLC6A6- is a knockout of that taurine transporter. So that means they can't take up taurine. And then they've got another condition. So the red is that mutant, that's what we're going to call it, the mutant. The green is the mutant plus the addition of senolytics. So they've added a few different compounds, which senolytics are a way to reduce senescent cells. And then they've got their wild type plus vehicle. So the wild type are fish that have not had this mutation. They have a perfectly intact taurine transporter that allows taurine into the cells. And what we can see here is, let's look at the longevity. So how long do these fish live? Clearly, the red is in real bad trouble. Once you knock out this taurine receptor, they don't live for very long. The maximum is 27 months. The next one, the green, is helped by the senolytics. So the senolytics do have an effect in reducing senescence most likely, And that does seem to extend their life by, let's say like three months, something like, because it doesn't quite reach 31. So the maximum lifespan extends by like 3 months. Now, of course, we're talking about fish, so 3 months is a pretty decent amount of time. Now, the wild type that do not have this mutant, so they can still accept taurine just like they normally would, they get this huge boost in life. So these are extremes. You're not going to have a taurine transporter deficiency or anything like that. But the point is that they're creating these extremes. If you remove taurine out of to be functional, you're in big trouble. So having taurine readily available can extend life quite substantially. And this would be kind of the normal lifespan that you would normally expect. Now, another condition that they could have added is to supplement with taurine and see how much further that extends life. But we already saw some of that data earlier in mice, which is far more applicable. Okay, so then another one is to look at this SA beta galactosides. Again, this is a measure for senescence. So if we see more blue, then we see greater senescence. Now here, and I should have added this because this is in my next slide, but I should have added this here. The TERT++ and TERT-, are, TERT++ is a enzyme that is called telomerase. So you may have heard of the fact that our DNA have caps at the end, which are essentially just these non-encoded sections of our DNA to essentially protect from the loss of DNA. So we have these caps at the end of our chromosomes that protect against when we replicate. So our cells replicate and divide. They have to make copies of everything from the original cell and replicate that 2 times so that they can create a new cell. So one copy stays with the original cell and another copy goes with the new cell. Now, during that process, we lose a little bit of the DNA that's at the very end of our genes, just because that's biologically how we're made. We have, every time we divide, we lose a little bit of the ends of all of our chromosomes. So what our body has, not in all of our cells, but in a lot of our cells, we have this telomerase enzyme, which will re-add or essentially add more of this cap to the end of the DNA. I'll show you an image of that in the next slide. I should have added it here, so I apologize for that, but I'll show it to you in the next slide. And this telomerase enzyme, if you don't have any and you keep dividing your cells, that means your cells eventually start eating into the actual DNA that's being read. That's a huge problem, because ultimately that leads, then the cell has to eventually become senescent because it can't replicate anymore because it's starting to eat into its encoded DNA. And you don't want to create another cell that also is eating into its DNA. So then these cells become senescent. And TERT here is, the TERT++ means that these fish do have functional telomerase, this enzyme that adds to the cap. TERT-, is deficient. So they have a mutant, they've generated this mutant that does not have telomerase. And we can see the comparison, the amount of blue in this fish compared to this fish is astronomically higher compared to this fish. So here's our control, here's our mutant, and then what they want to find out is, do we see a reduction with the addition of taurine? And we can see it's TERT-, it's the exact same condition as this, but they've added 300 micromolar of taurine, and they found that there is a reduction, at least by my eye, there's a reduction in the blue across the body. And the same is true for a much higher dose of taurine as well, but probably not much of a difference between these two. So a low dose is sufficient to see this effect. And we see that with 10 millimolar, which is many folds higher, we still see this effect, which I suppose isn't that shocking. Okay, so as I mentioned, here is an example. We've got our chromosome. If we were to pull a thread of that chromosome and just look at the DNA, we'd see that there are these caps with telomerase coming in and adding to the caps to make sure that the caps don't shrink too much. And here we're looking at beta-galactosidase intensity, which is that same senescent marker. And we see that we've got our TERT, so I kind of cut off the T there, the TERT plus plus, which means that telomerase is present, TERT minus minus, which indicates that it is not present. And we've got taurine not being supplemented, taurine not being supplemented, taurine being supplemented, and taurine being supplemented at different doses. And we see that when telomerase is present and taurine is not supplemented, that there is no issue with beta-galactosidase. It's very low. It's essentially zero, so there's very little to no senescence. However, when you have the knockout, so this is the first condition with the knockout, and no taurine, then you see this huge spike in beta-galactosidase, which is that marker of senescence. Then with the TERT knockout, so the telomerase knockout plus the addition of taurine, we see dramatic drops in the level of beta-galactosidase. So taurine has this substantial effect on cellular senescence, at least according to this model. Then they look at survival, and this is looking at this knockout of telomerase. We see that the knockout of telomerase is in red. We see that there is a dramatic drop. And then we see that with the addition of taurine that we see less of a drop, or it happens later in life. Then here we're looking at the DNA damage. So how much DNA damage is occurring, which is this marker. And we see that with taurine supplementation, there's a slight reduction in DNA damage. Now, the effect is noticeable, but it's not huge, but still does seem to be an effect. The next area that they wanted to look at was to look, what if we actually actively poison these animals? So if we actively add a poison, a toxin that a lot of people like to talk about, people love to use the word toxin. Well, this is an actual toxin. So they've added paraquat, which I was not familiar with what it was, but I looked it up, and it's a herbicide, and they're adding it to. So the red condition is the animals that are fed this paraquat and nothing else. So they're just fed, they're fed their normal diet, but like on top of their normal diet, they're also fed paraquat on a daily basis. Or I suppose in this situation, they're poisoning them just over like a few days. So we're talking about hours here. So 24, 48, 70. So yeah, just a few days. So like, let's say 3 or 4 or 5 days. And they can see that these animals do succumb to the paraquat quite readily, which isn't a shock. If you feed something poison, eventually it's going to die. On the other hand, what's really intriguing, and it's kind of a cool experiment, although I feel bad for the animals, is that the addition of paraquat plus taurine, you see that there's a delaying effect. So if you're afraid of being poisoned, then taking taurine supposedly will protect you for a little bit longer before you end up passing away. So we're only talking about hours difference, but it's still a cool effect to be able to say, okay, we supplement with this other supplement. It actually has a functional difference on our ability to, I hate this word, but detoxify toxins that are taken up by our body. And then the blue is the vehicle, which means that they're not given paraquat and they're not given taurine. So obviously none of those animals are going to die over the next 5 days. Another area that they wanted to look at was look at methylation status. So our DNA are wrapped up on these proteins called histones. And the histones can be opened or they can be closed. And that opening and closing is based off of these modifications, these epigenetics that are added to the histone as well, or called post-translational modifications in that situation, but let's just roll with it. Epigenetic modifications that are added to the histones as well as to the DNA itself. So the proteins here are the histones. We've got these tags that are added to the histones, and then we've got these tags that are added to the actual DNA string itself. And that would all be found inside of this chromosome. So if you were to pull on string, this is what you'd see. And then if you zoom in even further, and you actually see the actual DNA pairing the helical structure that people are used to seeing. So they're looking at the methylation difference between animals. So in skeletal muscle, in the cerebral cortex, the brain, and the liver, they're looking at aged, I think this is in mice, so aged mice, young mice, and aged plus taurine mice. So AT is age plus taurine. Now, I don't know why they did this exactly, but for some reason they switched these two. So here we've got the young, here we've got the aged, and here we've got the aged, and here we've got the young. Not sure why they switched that up, but it doesn't really make a difference in terms of reading it. You just have to be aware of it. And what we find is, so how to read this? I guess I should explain that first. The more purple it goes, the more methylation in that area. Does it matter what area? No, it doesn't. So just look at it generally. Just unfocus your eyes and just look at it as a whole and compare this column versus this column versus this column. Just look at the colors. That's it. That's all you have to do. Just look at the colors. And a light blue would be a reduction in the methylation status. So we're looking for similarity between specifically the aged on taurine and the young. Now, if I'm honest with you, I don't see that much of a difference. Like I see a dramatic difference still between the aged taurine and the young. You may disagree. One area that's slightly similar here in the skeletal muscle is this area right here. And you can see, but there's also a little bit of similarity in this area versus this area. So, but then there's a lot of dissimilarity between this area and the aged in this area. So I think that the way I would interpret this, is that it's a little bit of a middle ground between the young and the aged. It's not a complete reversal back to what the young animals are experiencing, their young methylation status. So is this massively like, wow, this is incredible kind of news? No, it's not. I think it's a little underwhelming, but it still provides proof that there's some difference, that taurine supplementation seems to have some sort of an effect. And you can do that for the cortex as well as the liver. Now here they're looking at the actual protein measures. So this H3K9 methylation 3, these are all different histones and methylation points. We don't need to know all about these different histones. All we need to do is essentially look at the comparison. So minus taurine in liver, brown fat, and muscle. So these are protein measures quantified by an experiment called a Western blot. And then the taurine, so on this side. So if you just look at this top one real quick in liver, you see that the methylation status of H3K27, a histone, is reduced in taurine compared to when the animals are not supplementing with taurine. We also see in this histone, we see also a massive reduction in the methylation status. On the other hand, when we look at brown fat, there does not seem to be much of a difference at all in this histone. So again, the same one as this one, but there is a slight reduction in the H3K9 methylation status. Then H3 here is, I'm assuming it's a loading control. So for anybody in cell biology and molecular biology, you'll know exactly what I'm talking about. Now, on the other hand, looking at muscle, we see that there's a shift from a reduced methylation of this H3K27 to an increased methylation. And here we also see an increase in methylation. All we're doing is looking at the size and the darkness of the smudge. That's it. And the loading is pretty good. Maybe this one's a little bit lower, but ultimately it's probably not going to make a huge difference. Now, to look at this again, we're looking at the exact same thing. We're looking at Western blots, and we're comparing the two conditions. So minus taurine plus taurine, minus taurine plus taurine for brown fat, liver, and muscle again. Here we're looking at the RS6P, so phospho RS6P, which is a downstream protein of mTOR. You may know mTOR. mTOR is a molecule that allows for essentially cellular growth. It allows for protein synthesis to occur and therefore eventually, potentially, cellular growth and proliferation, or the division of cells. And downstream of that is this RS6P. So this particular protein is activated by mTOR. And what we find is that with taurine, we see a complete loss in the phosphorylation status. Phosphorylation status in this instance is that if you phosphorylate RS6P, then you get more activity of RS6P. So you're activating RS6P to then undergo protein synthesis or or lead the genes to be read and to produce proteins for cellular growth. So you see a loss in the phosphorylation status of RS6P, or not complete loss in the liver, not a complete loss in muscle, but it's certainly a huge decrease in the amount of phosphorylation. So you're essentially shutting down this pathway, which is just one mechanism to activate mTOR. I just threw it in here. There's other mechanisms like amino acids specifically, or energy, or ATP and stuff like that. But I just wanted to throw this in here real quick. So you don't have to read all this stuff. Just know that mTOR gets activated, then you get a phosphorylation of RS6P. And in this situation, we're seeing that there is not that happening. So something along the line, that taurine is causing a stoppage of this mechanism. And then, so that's looking at phospho-RS6P versus the total RS6P. So the actual protein versus the activated protein, which is the phospho. And we see dramatic decreases. Okay, great. And then the other measure is to look at LC3A and B, so also known as LC3 1 and 2. And LC3 is a protein that's involved in autophagy. So as it gets cleaved, as it gets converted from LC3-1 to LC3-2, that means you have a more maturation of the autophagy pathway. And we read this by looking at this top band and seeing how much is cleaved to this bottom band. So what we see is that without taurine supplementation, we have a lot of LC3-1, so kind of the more immature version. And then there's a little bit that gets cleaved to the more mature version, but not much. On the other hand, here we see more that's cleaved into LC3-2. And the same is true for this. Looking at in the liver with the taurine supplementation, we see that there's an increase in the cleavage to LC3-2, and definitely the case when we look at muscle. So this would imply, we would have to see more data than this, but this would imply that you see increases in autophagy with taurine supplementation. All right, so let's look at some cytokines. So these are all, I'm not going to go over every single one of them, but we're looking at cytokines. We're looking at specifically inflammatory molecules that would either attract or inhibit the recruitment of immune cells. Now remember, before when we looked at immune cells, we saw that there was a phenotype shift with the higher taurine supplementation. So if we return here to the actual molecules that attract those immune cells or detract or inhibit those immune cells, what do we find? So again, I'm not going to point out all of them. Let's just focus on kind of the main ones. Let's just focus on like TNF alpha. You can read all these for yourself. So for TNF alpha, we're comparing the young versus the aged on the vehicle. So this is a placebo, and then the aged on taurine. So these are the same age, but one group is fed taurine or supplemented with taurine. So we see that so with the aging without taurine, we see an increase in TNF alpha, which is typically pro-inflammatory, typically. Now with the taurine supplementation, you do see a drop in the TNF alpha levels. They may not quite get to the point where they're as low as the young. I don't even know if they did statistics on this. I guess presumably they did, but there's no difference between these two. But I would probably argue that the effect size would lead to, there's probably still elevated TNF alpha. It's just not as high as the aged mice. So the point is that there is a drop in TNF alpha. And then if we look at some of these others, for the most part, there's no difference for some of these, which is really, really interesting. Like EO here, we have no difference between the young and the age, but then when we look at the age with taurine, we see a drop, even compared to the young. Really, really intriguing. And then you have some of these that don't have any sort of difference. So there's a lot of heterogeneity in terms of the effects, but overall, the way I would interpret this is that you see with taurine supplementation, you see a reduction in pro-inflammatory cytokines. Okay, let's get into stem cells. Then we're going to go into a little bit of human data, and then we're going to go into our midpoint conclusion from there. And then for the insiders, if you're continuing on, then we'll, in the full version of this video, then we'll also go into more human data and dosages and stuff like that. Okay, so for stem cells, what are we looking at here? So stem cells are cells that literally have unlimited potential. They can turn into any cell that your body needs. So if your body needs more red blood cells, then it will tell a stem cell, hey, start programming your genetic makeup to be more like red blood cells, and it'll start turning into a red blood cell. Or if you need an intestinal cell, it'll say, hey, stem cell, turn it into an intestinal cell. It'll start reprogramming itself into an intestinal cell. Now, there's this gene that's known to be related to mitotic activity of stem cells, meaning the cell division of stem cells, the activity of stem cells. So the more of this gene activity that you see, or the more of this gene in general, that means that you have more of these stem cells. So LGR5 is the gene, and really the protein that we're most interested in. So what they do is they do an experiment called an in situ hybridization assay, where they unwind the DNA inside the cells, and then they add a probe and they measure, and then that probe binds to specifically the LGR5, and it only binds to LGR5. So if you have a mutated version of LGR5, or if you have LGR3, it does not bind to that gene. It only binds to LGR5. So then that gives you a quantified amount of LGR5 that's found in a particular tissue, and then presumably stem cells. You get an idea of how many stem cells are there. And this probe also has a fluorescent tag attached to it. So then we can look at it under a microscope, the tissue itself, and be able to tell how much LGR5 is there by looking and quantifying the amount of fluorescence that comes off of an image. So here we've got the minus, so no taurine. Here we've got plus with taurine supplementation. We're looking at cross sections of different tissues. We've got gut tissue, we've got intestinal tissue, and we've got skin tissue here. And we're quantifying the amount of essentially pink, because the pink is the fluorescence that's coming from the probe that's attached to LGR5 specifically. And we find is in the gut, there's very little of this pink. With taurine supplementation, there's much higher levels of this pink. In skin, we see certainly more of the pink that's available, but when we supplement with taurine, when these animals supplement with taurine, we see much higher levels of this pink. So this indicate that there's more LGR5 in the taurine, therefore more stem cells. Now, this is not foolproof. We would actually need more data than this to actually really substantiate if there are more stem cells in general and if they're more active. But it's still good proof starting to point in that direction. So cool. Reactive oxygen species, you're probably well aware of that. They're damaging molecules that can damage all the inner sections of the cells and the surrounding cells. We see a drastic reduction in reactive oxygen species production. We see protein carbonylation as well as lipid peroxidation. These are two different markers of, again, of oxidative stress coming from reactive oxygen species. So if you see greater carbonylation, greater peroxidation, that means that the fats, so for lipids, and protein carbonylation is for proteins, obviously, you get more damage of those proteins. Remember, these molecules have functions inside the cell. So if you have more of these damaged molecules, your cell has to then recycle those and get rid of them. So the less of that you can have, the better off you are. The cell doesn't have to do as much of this recycling process. So, and then you have more of these functional proteins as opposed to non-functional proteins. So the protein carbonylation, we see that there is a drop with the taurine supplementation, that's AT, and with peroxidation, we also see a drop. The effects aren't huge, but they are present. Hey, if you're interested in having greater access to me, being able to ask me questions, as well as the optimal dosing for taurine, as well as a slew of other videos that actually have more applicable takeaways, and you can join live sessions with me and things of that nature, as well as truly a whole bunch of other benefits, then consider joining the Physionic Insiders, which is a smaller community, a smaller program where I release content on a weekly basis, and I also have every month or so these live sessions where you get to ask questions of me, and then I do a little bit of research, a little bit of background on that question, and then answer it live for you in kind of a loose format, and then you can ask follow-up questions as well. So if you're interested in joining that, then certainly consider joining in the link that's down below in the description box or in the pinned comment as well. And now let's get back to the public version of this study analysis. Okay, now let's get into just one piece of human data. And here we're looking at, so it's a separate study, Epic Norfolk studies, a separate study from this particular study, but they're using the data that they've taken back, I think this was back in like the 90s or early 2000s. They had all the data sets. So the researchers took those data sets and then reanalyzed them specifically for taurine, because at the time they weren't looking at taurine. But they had the data that included taurine, hypotaurine, and N-acetyltaurine, and these are metabolites of taurine. I'm going to focus on taurine, but if you're interested, I decide to add them anyway. Hypotaurine and N-acetyltaurine, again, just metabolites of taurine. So this is according to blood values of taurine. If you see any one of these measures going more blue, then you see a reduction in this measure with increased taurine. So that's an inverse relationship. If you see more red, then you see higher levels of that particular marker. So let's look at some of these. So abdominal obesity, it's slightly bluish, means that it's starting to reduce with higher levels of taurine. So that inverse relationship. The same with obesity. You see a slight reduction in obesity with higher levels of taurine. Now, I need to emphasize this. This is pure association. This is just finding a relationship. It is not saying that with increased taurine, you see reduced obesity. All they're trying to do is create a correlation in humans, and then your mind would go, oh, well, did we see some of that in the actual causal data looking at the animal studies? And we did see that, right? We saw that there were reductions in fat mass, not that taurine specifically caused fat loss, but it protected from fat gain compared to the animals that didn't consume taurine. Another area would be like looking at diabetes and glucose. We see reductions, and specifically really strong reductions in diabetes when blood taurine levels are higher. Now, here's where I really have to caution you, because this is something that the researchers actually don't do a great job pointing out, which is I was kind of surprised by. I wish that they had pointed this out more, is if you look at ApoB, if you look at cholesterol, if you look at dyslipidemia, if you look at hypercholesteremia, if you look at LDL, those are all slightly elevated with increased taurine. So now we're looking at a direct relationship as an inverse instead of an inverse relationship. We see that there are these, there's this relationship with increased levels of these. Now, again, it's not causative. It could be because of something else that also correlates with taurine, but we don't have the data, and they didn't have the statistical ability to start to tease out some of these effects. So this is just pure association with no meta regressions, no multivariate analyses, none of that. It's just pure, hey, if taurine goes up, then what happens to this? That's it. So this is really intriguing, but certainly keep an eye out on this. Am I worried by this? No, I'm not. I'm not worried by this alone. I would need to actually see direct causative data to see, oh, if you supplement taurine, do we see increase in ApoB, for example? I would actually, and we can test that. We can actually see that. So that's something I plan on doing in the future. Okay, so let's kind of wrap this up for the public version, and then we're going to continue on with the insiders version of this video. So for the midpoint conclusion, the taurine supplementation had drastic, wide-reaching effects in multiple model organisms. We're talking about mice, monkeys, fish, worms. The effects extend longer life. They improve metabolic measures, which I'm just going over just a few of them. There were a whole lot of different markers, including bone and stuff like that, and muscle, blood sugar, body fat reductions. They improved markers of cellular aging, so specifically senescence. We saw some robust data on that. Changed the inflammatory profile. We saw that changes in tumor necrosis. Tumor necrosis factor alpha. Excuse me, that like for some reason my brain was like, is it factor necrosis? I don't know. Anyway, my brain just went kind of haywire there for a second. So changed inflammatory profile. Also, we saw changes in the immune cells, white blood cells, monocytes and granulocytes based off of really the higher taurine consumption, not as much with the lower taurine consumption. Better muscle and bone strength, which I mentioned, among many other benefits that you'd have to go back to actually watch the video, looking at DNA, methylation, all that stuff, DNA damage. The human data is associative, but indicates that blood taurine levels, as they increase, there's a relationship with reduced diabetes risk, reduced obesity, and reduced inflammation. However, there's also a relationship, an association with increased dyslipidemia as just one example. But we actually need more human data to actually create a causative link in that situation. Now, if you're interested in learning more on that human data, if you'd like to know the optimal effect of dosing in humans specifically, and you'd like to know a really weird, cool experiment that they did in relation to exercise and its effect on blood taurine levels, and how that might relate to our overall health, then certainly join my Physionic Insiders program, where you'll get access to this video as well as all my other videos, full study analyses with applicable takeaways and all that good stuff. But if you're ending it here, then certainly I welcome you to watch some of my other public content. And thanks for stopping by. I really appreciate your patience. Hopefully you learned something. And yeah, it was great talking some science with you. I'm looking forward to next time. Have a good one. Bye.